My name is Thomas Riley Morse with Hazen and Sawyer, and I do a wide variety of work, both water and wastewater, and I have interest in biological processes and disinfection.
Awesome, thank you. All right, so let's go ahead, move on. So this project relates to landfills who are collecting their leachate and storing the leachate in storage ponds. And in Florida, sorry, I have like these weird things going on. I don't know how to turn my timings off. But anyways, in Florida, there are two main options for a landfill to dispose of their leachate. So they can either send their leachate to a wastewater treatment plant, or they can depot inject their landfill leachate. So this project relates to those landfills that are sending their leachate to a wastewater treatment plant. And there's anecdotal and also data evidence that the disposal of landfill leachate, the introduction of that into a wastewater treatment plant, can cause some upset. So one potential upset is ammonia removal inhibition. There's also biological treatment upset, and then just plain old overloading into specifically the activated sludge process of wastewater treatment. So down here in Florida, landfills are charged, at least in Lee County, landfills are charged for different constituents that may be in their leachate. So for example, if you look at chemical oxygen demand here on the slide, a landfill would be charged for their leachate over 600 milligrams per liter of COD. And they're going to pay $9.78 per 100 pounds of chemical oxygen demand over 600 milligrams per liter, up to a chemical oxygen demand maximum of 6,200 milligrams per liter. So that's difficult because leachate can range from 50 to 90,000 milligrams per liter of chemical oxygen demand. In our study, the leachate that we've been using, the COD averages between 6,400 and 6,800 milligrams per liter. So there's this motivation to try to reduce constituents like chemical oxygen demand to try to save cost of like disposal costs. So another thing we could look at is TKN. So this nitrogen, landfills are going to be charged $158.70 here in Lee County for 100 pounds of TKN over 30 milligrams per liter. And leachate can range all the way up to almost 2,000 milligrams per liter. So there's motivation to pre-treat this leachate before it's disposed to a wastewater treatment plant. All right. So this is an image of a leachate storage pond in Florida. And I went and visited this particular landfill. And it was really interesting because they had, sorry about the timings, they had this attempt at floating some plants onto this leachate pond. And the plants were dead, and it didn't really work out. But it got us into this conversation about, well, what if we could have some plants that could survive the harsh conditions of floating in leachate, and these plants could uptake some nitrogen, they could reduce the chemical oxygen demand, and therefore pre-treat the leachate before being disposed to a wastewater treatment plant. So that's the idea behind this project. This is like a funny little graphic, but it gets the message across. So what we would like to do is just float some floating treatment wetlands on top of our leachate storage ponds. So that's the idea behind the project. And clearly the main challenge first is just to find some plants that can actually survive floating on leachate. So our research questions here, first off, can we identify 3 plants natural to Florida that can tolerate living in a leachate pond? So I'm using natural here kind of loosely. So I don't mean native, I mean natural. They can grow, they're happy to grow in South Florida. So for example, water hyacinth is extremely invasive down here. But water hyacinth is the only plant that's been researched that can survive floating on leachate. So the background research that we're working off of is water hyacinth can survive floating in landfill leachate and can reduce the nitrogen and chemical oxygen demand loading of landfill leachate. So water hyacinth would be very difficult to use in Florida here. In fact, like we can't even get water hyacinth for our lab-based project without having a permit for water hyacinth. So it's just so invasive. So that's not really going to be ideal for landfill leachate storage ponds here in Florida. So the goal is to find 3 plants that can do it besides water hyacinth. The second question we have is, once we can identify at least 3 plants that can survive floating in leachate, what is their ability to reduce chemical oxygen demand and total inorganic nitrogen? And we're going to be testing this in bench scale batch reactors. The third is, what effect does dissolved oxygen and pH have on the phytoremediation treatment efficacy? So if we were to adjust the dissolved oxygen or pH of the landfill leachate, how would that affect the treatment? And then lastly, does it make sense to co-treat septic tank effluent or some other wastewater discharge that would allow the leachate to be diluted a little bit to make the liquid more friendly for plants to survive in and therefore improve the treatment efficacy? So kind of want to go through each of these questions specifically. So the first one is just finding some plants that can survive on leachate. So the objective is to use bench scale discontinuous batch reactors with landfill leachate from a closed class 1 landfill to identify at least 3 saline-tolerant floating leaves or emergent plants natural to South Florida that can survive on a floating wetland in a landfill leachate storage pond. So you can see just some photos here. The idea is just to have a little batch reactor filled with leachate and to float the plants in them. So we fill these glass containers with leachate and then leave that leachate with the plant for a certain hydraulic residence time and then just take it all out. So it's a discontinuous batch reactor. So our original thought was to set up about 10 of these tanks and fill each one with a different plant. So you can see the original idea was to use water hyacinth, which is extremely invasive down here. Water lettuce is also invasive, and then a few other plants that are happy to grow in South Florida. So that's what we set out to do. Obviously, we found that water hyacinth requires a permit, so that's a little more complicated. But we've been working with some local nurseries and also ECHO Global Farms, who work in developing countries to come up with solutions for food security and also to improve drinking water treatments. This is a really cool organization down here. They do a lot of agricultural work. So we've been able to procure some plants from them and also from some local nurseries. So currently we actually have 20 different plants that we're going to be testing. So originally we thought we would do 10 plants that we would test and hopefully find 3. So now we have 20. So that's really exciting. So you can see we have this huge like metal trough filled with really cool plants. So we're going to be testing all of these and their survivability in landfill leachate. So what you can see here is just another glimpse of our setup. So what we have is in this big metal trough, that's mimicking a wetland. So we have 8 inches of soil and 2 inches of standing water on top of that soil. And the idea behind this metal trough is just to grow the plants, just to see if we can just get them to stay alive in soil, and we're watering them with lake water. So the first thing we need to do is just keep the plants alive outside in our lab, and then we can expose them to leachate. So in the metal trough, there's no leachate. We're just trying to keep the plants alive with our setup. These 2 plastic containers that you see towards the bottom of the picture on the left-hand side, these are just filled with lake water. There's no soil. Well, there's a little bit of soil at the bottom, but like ½ an inch of soil just to provide some nutrients. So these plants in these plastic containers are floating. The ones that are in the metal trough, they require a little bit of substrate to have their roots in to survive. So the ones in the metal trough, plants that need soil, but are happy being very saturated with water. And then the ones in the plastic containers are actual floating plants. So what we've done is we have this setup here where we have all of our plants growing just in these bins and then the metal trough. And then the leachate challenge tests are happening in these glass reactors here. So again, the blue arrows are pointing to where we're just growing the plants. This is the plant cultivation. We're just growing plants in soil and lake water. And then the glass containers that you can see in the secondary containment of the plastic bins, that's where we're having these leachate challenges. So we're only 6 weeks into our project, but we've done preliminary leachate testing already on 5 plants. So I'm going to show you what we've done so far. So the first one that we decided to work with was Asiatic pennywort. And instead of just putting these plants straight into landfill leachate, we decided that it might be interesting to see, well, what if we just do 25% leachate and 75% lake water, just to see where's that breaking point. So the idea is we'll do 25% leachate, then we'll put them in 50% leachate with 50% lake water, and 75% leachate, and then 100% leachate, just to see maybe some plants can survive the 50% leachate ratio, and then we can explore, well, why is that? So this is Asiatic pennywort, and you can see on day 0, it was nice and okay, floating in this leachate, 25% leachate, 75% lake water, and we've taken pictures for 7 days, and it's still doing okay. So you can see some of the leaves are yellowing a little bit, and that's maybe showing signs of some toxicity, but it is still alive 7 days into it. So that's good. This is we're relatively optimistic about this Asiatic pennywort to date. This is in a nice contrast to this water lily that we have. So this water lily, we forgot to take a picture on day 0, but day 1, you can already tell it's kind of unhappy. You're seeing signs of toxicity, just really not doing great. Day 2, it's really just kind of, I don't know, dying, just straight up dying. And by day 5, it's completely disappeared. It's just dead, big ball of dead mess in this liquid here. So water lily is super unhappy with our setup and the leachate that we're using. So that's not gonna work. We have 3 other ones that we've looked at. So elephant ear looks fine. So day 0 to day 7 looks basically the same. It even has some new growth happening. We have a new leaf that's popped out. So it's all right. So we'll move this elephant ear plant to a 50% leachate ratio. And then if it can survive that, we'll move it to 75 and then 100%. And that'll be really exciting. Water hyssop, we're seeing similar results. So it seems fine after day 7. And then Echo, Echo Global Farms, the one that do really cool research on food security and things like that and international development. They gave us this plant and didn't tell us the name of it. So I think it's related to alligator flag, but we're still waiting back to hear from them what is this, because it does seem to be okay. I do just want to explain the color change here from day 0 to day 1. So this plant needs to be rooted in substrate. So we put some sand in here on day 0. And then we realized the sand was a little bit too fine. So day 1, we added some actual soil, like some compost type soil with some pebbles to keep it, you know, in the upright position. We don't want the roots floating up and the leaves going down, because then it's just going to die because it's unhappy with its stance there. So anyways, the difference between day 0 and day 1 is just we added some soil and rocks. So that's the color difference you're seeing. But this mystery plant seems to be happy. So we'll hear back from Echo about what exactly this plant is, and we'll move it forward with 50% leachate testing. So this is what we've done with Objective 1 so far. We've tested 5 plants and 25% leachate. So we have 15 plants to go and several more dilutions to go. So we're working all of these plants up to a 100% leachate solution to see if it can float in a leachate storage pond. So that's the idea there. I kind of just want to stop here to see if anyone has any questions.
Yeah, I had one question. Are you using anything to measure the actual plant growth, or is it all visual observations?
Yeah, good question. So for Objective 1, we're just using visual observation to see if the plant can survive. Once we get to Objective 2, we're going to measure biomass change and also, you know, the reduction of pollutants within the liquid. So we'll measure chemical oxygen demand and ammonium and things like that. Does that answer your question?
Yes. I had one more question. How are you growing all these? Are they in a greenhouse, or do you have grow lights set up?
Right. So the original idea was to have grow lights inside of a temperature controlled lab, but we have some lovely outdoor space with natural sunshine. So I've decided to move them all outside just to save energy and save the setup here. But that's definitely open for dialogue if that needs to be adjusted. But currently everything's just outside.
Thanks.
And
Right.
this is Thomas. One quick question. As far as the controls, I'm assuming you're doing like a set that's just exposed to like normal, just regular irrigation or water.
Yeah, so let me go back. These plants are just grown in soil and lake water just to keep them alive. We don't have, you know, specific controls as far as plants floating in lake water into glass containers. We don't have that at this time because the goal here is just to find something that can survive. But, okay, you know, I've thought of that because what if they're dying just because the slide timing is annoying me? Sorry. What if they're dying just because, you know, that's the setup in the glass reactor? So that thought has crossed my mind. So we could definitely be doing controls in Objective 1. But again, the whole goal of Objective 1 is just to find something that can survive in our setup. So.
Okay, no, that sounds great. Awesome, thank you.
Okay. All right, great. Any other questions or comments on objective 1.
So what was the hydraulic retention time, residence time? So I believe the leachate and the lake water comes in and goes out as a gallons per minute or some flow rate, right?
Well, in the leachate storage pond that we're kind of modeling all this after, there's a certain hydraulic residence time, which I don't actually know that at this point. So we're going to confer with the landfill when we, once we identify something that can survive in landfill leachate, then we'll model our hydraulic residence time after their storage pond. For this first objective, there's no continuous flow or anything like that. So these pictures where we have day 0 to day 7, that's the same water for all 7 days. There's no flow in or flow out.
So well, let's say day 7. So you have still in this picture, you see the plant is still growing. At least you have a profile of reduction on water quality, or you haven't done yet.
No. So objective 2, which we haven't discussed yet, but objective 2, we will be measuring the change in water quality. Yeah. Okay. So this first objective, we haven't looked at the leachate at all. We're just trying to figure out things that can survive.
Yeah. One last thing, on one of your slides, you said the TKN or ammonia is 2000, but in the slide it shows 200. Maximum. Typically, I have seen in leachate, ammonia can go up to about 3,500, but I think maybe you meant when you spoke, you said 2,000, but it's here in 200 maximum.
In the blue, leachate can range up to 2,000 TKN, but I agree with you. Ammonium can be, yeah, maybe up to 3,500. So that, obviously would mean the TKN would go up to at least 3,500. But the 200 milligrams per liter that you see here written in white, that is what the wastewater treatment plant will accept for a industrial wastewater flow into their wastewater treatment plant. They will accept up to 200 milligrams per liter of TKN in that flow, which is a problem if your leachate is over 200, because then they won't accept it.
Oh yeah, okay, I got it.
Right. Okay. Any other questions on this slide or objective 1, or any suggestions? Do we suggest measuring, I don't know, the change in the leachate quality at this point? Because I think that'd be interesting to see, but let me talk about objective 2 to kind of address that. So our second objective is, once we can actually identify plants that can survive floating in leachate, which, I think that's our biggest challenge at this point, because a lot of plants are just not going to be happy floating in landfill leachate. But once we can identify them, then we'll put them in the same setup, floating in the little glass batch reactor. We'll have our hydraulic residence time and measure the change in leachate quality over that time. So once we know the plant can survive, then we'll start looking at the leachate quality as far as chemical oxygen demand, ammonium, nitrite, those kinds of things. So this objective, not only will we be looking at the plants in pure leachate, so we'll be doing that in duplicate. So you can see here, reactors 1 and 2 would be plant 1 in duplicate. So that would be, nice duplicate data there. Reactor 3 and 4 would be the same plant, but modeling a wet season situation. So down here in South Florida, we're quickly moving into our dry season. So the leachate that we'll be collecting for the study will be dry season leachate. So the idea is that we would dilute it a little bit to model what that leachate would look like during wet season and how that might change the removal efficacy of these different pollutants that we're interested in. So objective 2, we'll use the plants that we identified that can survive. We'll measure the change in leachate quality and as far as chemical oxygen demand, nitrogen, and then the various other constituents that we're looking at. And we'll model dry season versus wet season. So any comments or questions about that?
Yeah,
Go ahead,
just
Ryan.
one thing that kind of raises a little red flag for me right now is when you're dealing with plants, sometimes duplicate is not enough, because you're just going to have a lot of variability in the plant itself to begin with, let alone seeing any impact of the treatment. And when I ran the greenhouse studies when we were both at Duke, I had quadruplicates for every treatment. It ended up I had to reduce the number of treatments I did, but I feel I had a better representation of what was going on.
Yeah, I think quadruplicate is definitely doable here. So yeah, we can look at that for sure. Thank you. Any other questions or comments? Okay. So objective 3, the idea here is that we would adjust the dissolved oxygen and pH of the leachate that we have to see how that might affect the treatment efficacy. So the idea behind that is you can see this is the same leachate storage pond that I showed you at the beginning of the presentation. And you can see this right here is leachate just coming out of these pipes and slowly filtering into the pond. Well, there's this precipitate that's coming out of the leachate. So we're now just referring to this as leachate rocking. And in our landfills down here, you know, a lot of our different landfills, they're experiencing blocking in their pipes from this leachate rocking, in fact. So as the leachate flows through, it just little by little is, you know, having this precipitate that comes out. This is, no, okay, yeah, we'll talk.
Yeah, we experienced that rocking up here in Volusia as well. Same exact thing.
Yeah. Well, not awesome. Yes, exactly.
Yeah,
so we're having
definitely not awesome.
No, not awesome at all. Not awesome. So we're having this leachate rocking, and so last year we had a project that was going on, and we were putting leachate into reactors via peristaltic pumps and really small tubes. And what we saw over the course of the project is we had precipitates coming from our leachate. So this is a bunch of precipitate at the bottom of our tank after a few months of being exposed to leachate. There's none in the middle because we had a stir bar, and the stir bar actually could not stir because the precipitate was so thick that the stir bar couldn't stir. And we don't have pipes, so we had tubing, and our tubing got clogged by leachate rocking, and we actually had a couple tubes burst. So the leachate rocking, big problem. But the idea behind this third objective is if you adjust the pH or the dissolved oxygen, does that impact the leachate rocking? Because the leachate rock itself, we don't know quite the composition, and it's possible that part of the leachate rocking could be some sort of saline chemical. So if we're precipitating out some of the salinity, that could increase the survivability of the plants. So the question here is, if we adjust the DO and pH, does that affect the leachate rocking? But either way, it could affect the survivability of the plants. So objective 3, we'll look at DO and pH changes.
Was that Juno that talked before? And if so, could you comment if they did any testing to see what that rocking was?
Yeah, that was Juno, and no, we have not done any testing on what the rocking is at this point.
Okay, thanks.
Dr. Townsend, right?
Well, yeah, so this is Webster with the Heat Leak Center. We fund, and we have funded, several projects on rocking, and I think, and I don't know a whole lot about it, but I think a lot of times it has to do with ash. So we have a lot of waste energy plants here. So if you're using, you know, ash in your, if you have a monofill or you're using ash in your daily cover, things like that, I think it leads to the rocking. Dr. Miroff is a good resource. Okay. Miroff and Dr. Townsend both would be great resources for that.
Yeah.
And remind me, Ashley.
Go ahead, Ryan.
And remind me, did you change the pH in your previous study where you saw that rocking?
I did not, no. We haven't done that. But there has been research done from other Florida universities.
Yeah, I was just curious to see if you had seen a difference if you had changed the pH. I couldn't remember.
Right. No, we haven't changed the pH or tested that. All right. And then our last objective would be to, so the original idea is, what if we dilute the leachate a little bit, and that may help improve plant survivability. So if we can improve plant survivability, we could improve the phytoremediation and the treatment efficacy of the overall process. So we thought about, what if we dilute with septic tank effluent? And the way that this idea came about is sometimes landfills are, you know, relatively far removed from the main municipal infrastructure, and they may be on the septic tank. And if that's the case, what if we could use that septic tank effluent in the leachate storage pond? So that idea, like, how practical is that really? You know, I'd really like to discuss that and see if you have any suggestions of what else we could dilute with. But again, the idea behind this is, what if it's diluted a little bit? Could that improve the plant survivability? And then we are suggesting performing a cost-benefit analysis. If you increase the volume that you have to discharge to a wastewater treatment plant, for example, then you're going to increase your cost of treatment, because the more volume you have, the more you have to pay. But if you can decrease the pollutants, you know, there's just a balance there of volume to pollutant concentration. So the septic tank, that's not necessarily applicable if we're discharging to a wastewater treatment plant, because if you can discharge your leachate to a wastewater treatment plant, your landfill is probably also hooked up to a wastewater treatment plant for the wastewater coming out of the office, for example. So just the idea behind this, what if we're diluting a little bit? So I'd really love to open that up to suggestions and changes, like, where can we take this fourth objective, if anyone has any ideas.
So when you dilute that with like septic tank effluent, so that then you have two types of reactions. So there is one uptake from the plants, and then also it's like a biological reactor, and the bacteria might anaerobic bacteria might take up some of the pollutants. So I believe then it is two reactors, I would say. I mean, one is a plants and one is a biological reaction. Is that what you are trying to do here?
Definitely. So any sort of bioremediation we can have with microbial communities would be excellent. My just general concern is, I don't know how well a microbial community will do in pure leachate, but you know, certainly that would be great if we have some microbial community surviving and working and reducing the pollutants. Definitely.
So
and
I think.
Go ahead. Mm-hmm.
Sorry, I think it will work well. I mean, Juno can also tell about his sequencing batch reactor. So he's introduced leachate to a biological community, and I think if you do it right with the right dilution, I mean, not like a shock loading, the bacteria will thrive on a certain conditions. Like you saw, some of the plants even die when you have certain dosage and certain plants. So it's the same thing. I mean, certain bacteria will die, but others will survive. And also you have to increase, I mean, dilution can be done step by step to see where they can survive.
Definitely, thank you. Juno, have you seen any, I guess you've been doing some treatment wetlands, is that right?
No, we're running an SBR plant. So we do a biological treatment to it, and then we discharge via a spray field. So we discharge our effluent to a big spray field.
Okay.
But one thing that we are looking at is potentially doing some wetland treatment on the effluent in the future. But I hadn't thought about doing it because we have the same style open ponds, and if we could get almost some pre-treatment before we ran it through our treatment system, may also help.
Okay. All right. Isaac, did you join the call? I think Isaac's on the call. All right. So
I thought hitting mute on my puck would work. I did join it. Hi, Ashley.
Hey. Awesome. Welcome. So if there's no suggestions for objective 4, then I think we'll probably just move forward as is, and we'll see how the different dilutions can affect not only the plant survivability, but hopefully some microbial treatment. So that could be interesting to see. All right, so our timeline, we have a year to complete this study, and so far we're on track, although now we have 20 plants to test, so objective 1 will probably continue to go on for a while, but objective 2, there's no reason why we can't, start objective 2 now that we've identified, well, we still need to do 100% leachate, but objective 2 will be coming along soon, where we're measuring, the treatment of the leachate and measuring those different constituents. So this will continue all the way to next August, and then a few weeks after that to do some report writing. And of course, the anticipated benefit is if we can passively treat landfill leachate with this low cost pre-treatment technology, that could be really great as far as reducing leachate management costs. So that's the overall goal, is to identify some plants that can survive, see what their treatment abilities are, and hopefully we can be pre-treating leachate before disposal to wastewater treatment plants and save money. And this is especially important for perhaps closed landfills that are on this really fixed budget. All right, so with that, any questions, any suggestions, anything like that is appreciated. So please, if you have anything else to say, go ahead. All right, Wes, is there anything you want to add? No? Okay. All right, well, if that's all we have. If no one has any comments or suggestions or questions, we're going to post this tag meeting to our website. We have a website up and running. I'll send that to you. And thank you so much for being on this tag, and we'll schedule another meeting in several months from now. So please reach out to me if you have any questions or suggestions as time goes on. So thank you so much, everyone.
Thanks a lot, Ashley.
Thank you.
Thank you.
Thanks, everyone.
Thanks, Ashley. Good luck.
Thanks. Bye-bye.